Welcome back to this channel. Please, if you're viewing for the first time, just make sure you subscribe and hit the notification bell. Today we are going to talk about erythropoiesis, and this is the process of red blood cell formation. Yes, we are going to see it. If you didn't view my previous video about the general overview of hematopoiesis, please view it. Erythropoiesis is a process of red blood cell formation, and from the bone marrow, as we said in my previous video, we have this cell, the hematopoietic stem cell. And the hematopoietic stem cell has capability of forming all cell type, but however, depending on the hormone stimulating the process, a specific cell type will be formed. In erythropoiesis, the major hormone is erythropoietin, and erythropoietin is produced by the kidney. When the hematopoietic stem cell gets instructions from erythropoietin, it just understands that this is now red cell formation, because erythropoietin comes with instructions like, hello Mr. HSC, we are going to form a cell that is going to be having a protein. So literally it comes with instructions of protein synthesis, and the protein is hemoglobin. The hematopoietic stem cell will be now stimulated to form progenitors, but for the red cell lineage this type, since it is erythropoietin stimulating the process. And these are the blast forming units, erythroblast, and the colony forming units, erythroblast. These progenitors are capable of producing over 200 blood cells responsible for forming red blood cells, and every progenitor is capable of producing that amount. So it depends on the progenitors available and those stimulated according to the need in the body. So the blast and the colony forming units, erythroblast, will be stimulated to form a cell called a pronomoblast. The pronomoblast will be the initial cell formed, and this is a large cell with nucleus that has visible nuclei. The cytoplasm of this cell is highly basophilic, and it has a nucleus, the pronomoblast. And this normoblast will then develop into an early erythroblast. It can also be called a basophilic erythroblast. Why is it called basophilic erythroblast? Because each cytoplasm stains basophilic, and this is due to the fact that since we know that hemoglobin is a protein, and in the process of protein synthesis we have stages. It comes from the DNA of the cell, it undergoes transcription to form the RNA, which undergoes translation to form the protein, which is HB for this case. And in this process of forming RNA, from DNA to RNA, this cytoplasmic RNA, it is a ribonucleic acid. And these acids have affinity for the basic component of the Romanowski stain, which is methylene blue. So the cytoplasm will be basophilic to look dark blue because of the acidic component of the RNA that is in the cytoplasm. Remember, it is this RNA that now will be forming the protein which we need in the cytoplasm as hemoglobin to make it a red blood cell. So that's why the cytoplasm is basophilic in stages of early red blood cells. We said this is the early normoblast, which can also be called basophilic erythroblast. This will give rise to an intermediate erythroblast, which is also called polychromatic erythroblast. It is called polychromatic because it now stains for both the hemoglobin and the cytoplasmic RNA. Remember, it is a process of protein formation. This will not come as a magic that the protein will come. It will go process by process, and we shall be seeing that the RNA available that makes the cytoplasm basophilic is reducing, as it stains also pinkish for hemoglobin that is being formed. Part of the RNA is being converted to the protein, which will show up as a pink stain. Then the residual RNA that is still in the cytoplasm will still make the cytoplasm a little basophilic, giving it two colors. So the intermediate erythroblast gives rise to a late erythroblast, which is also called orthochromatic erythroblast. It is called orthochromatic because now it almost stains as a red blood cell or a mature red blood cell. Ortho is a term to mean similar, or to put upright. It's like meaning upright or correct or similar. Something similar to the normal. So it is orthochromatic because it is almost staining as a mature red blood cell. Orthochromatic. What we need to realize about these cells is, as a red cell grows, the size of the cell is reducing. The pronormoblast was larger. The arenormoblast was smaller than the pronormoblast, but still larger than the subsequent cell coming. The intermediate will be smaller than the arenormoblast, but still larger than the late erythroblast. The late erythroblast will be larger than the erythrocyte. The cell becomes smaller as it grows. Also, that the nucleus is getting smaller and removed from the cell gradually. So our first cell had it larger. It became smaller as it giving out its DNA to be formed into RNA to form the protein we need. Then here, finally, the nucleus is removed from the cell, and at this point we also have removal of the mitochondria. Why are we removing the mitochondria and the nucleus? The nucleus its component is needed in protein formation, so it has to get going. Then the mitochondria is some good element of oxygen consumption, so we don't need it in the cell because the cell is purposely being made for oxygen transportation around the body. So the mitochondria is removed. It has a series of events in it that utilize oxygen. So the late erythroblast is now getting rid of the nucleus and the mitochondria. It is giving rise to a cell called a reticulocyte. The reticulocyte is formed within the bone marrow, and it is sent into the bloodstream through the bone marrow sinusoids. The bone marrow sinusoids are small veins that drain material from the bone marrow. So the bone marrow sends the reticulocyte into saturation, and in few hours of the day, the reticulocyte will develop into a mature red blood cell. which has its hemoglobin in it without the nucleus, and it is not basophilic in staining because the RNA is now all used up and we have formed our protein hemoglobin. This is now the mature red blood cell. Maybe a quick review of the process. You have seen the hematopoietic stem cell. You have seen the hematopoietic stem cell giving rise to progenitors of the red blood cell formation, burst forming unit erythroblast and colon forming unit erythroblast. These give rise to a pronormoblast, which is a larger cell with a nucleus, intensely basophilic cytoplasm, which gives rise to an arenormoblast or the basophilic erythroblast. The arenormoblast gives rise to the intermediate gives rise to the late erythroblast gives rise to the reticulocyte, which is eventually sent into the bloodstream, and after 24 hours, it's expected to have developed into a mature red blood cell without ribosomal RNAs in it, and has our protein, the HB. So here the cell is fit for oxygen transportation. It lacks a mitochondria, it lacks a nucleus, and it's without ability of this protein grasping oxygen and having it in a reversible way, because it has to capture it, then give it out to the tissue. Yes, that has been the process of red blood cell formation, and up next is white blood cell formation. Just make sure you subscribe. If you didn't hit the notification bell, please do. You'll be notified when I post a video. Thank you for watching.